Hi, Aravind. I thought this is a good time to sort of summarize a very long journey of yours and of which I have been a part now. Um, that goes back to about 25 years. I don't know if you remember when we met about 25 years ago in 96. Um, on the first day itself, when we met, we were discussing about apoptosis as such, and you already had some earlier notions of the process and some ideas of proteins that might be involved in it, and some basic ideas of kin selection. So I remember that conversation quite clearly, and we've come a long way from there to where we now, I think, have a solid understanding of the origins of apoptosis um, from some very recent works that we have published from our group. So I think it's a good time to sort of sit back and not only reflect on the journey, but also provide a perspective that we have in our group about the origins and history of apoptosis as a study and also like from a biological perspective.
Yes, Lax, I do remember that conversation. I think the focus 25 years back when we first met on that long drive from Houston to College Station um, was on the resistance genes in plants, which have an APA TPase, what today we call an APA TPase. And I think I was telling you of my earliest observations that I had found some bacterial homologs
Right, right.
which sort of was to set the journey or start the journey into this discovery of the origins of apoptosis. So indeed, it would be a good thing to reflect upon all that has happened in the past 25 years. It remains as exciting now as then. I do clearly remember the day. It was back in India in I think 95 when I was scanning the pages of the Journal of Bacteriology, if I remember right, and there was the report of the discovery of a streptomyces protein, AFSR, and it had an ATPase domain, which the authors did recognize. But it struck me that this ATPase domain had a relationship to the resistance proteins like N um, in plants, and that was, in a sense, the birth of the APA TPases, the stand superfamily, and many other things which were to play an important role in our understanding of apoptosis in the following years.
Right, right. I mean, yeah, I think there's this whole journey of discovery that I think we can talk about, and one thing that we can do though, which I really like, I think this is a very good avenue for us to do, is to sort of go step back a bit on to understanding why there is apoptosis at all, from an evolutionary perspective. What is the general biological context of this phenomenon that is independent of, for example, its presence in animals, plants, fungi, and so on, and now even bacteria. That is, what are the general principles for where such a system can evolve? Um, I think that will be a good perspective for us to get from you.
Yes, that's not a perspective which is often spoken of in studies on apoptosis. So I think that would indeed be something worth discussing for a more general audience. But right at the outset, I should state that whatever we are going to be talking about today would be presented from our perspective. It's sort of a personal perspective. It's not to demean others' contributions, but it's going to be a narrative like we telling our history. So I should just get that out of the way right away.
Right.
And my own interest in this began in a very different way, perhaps, from how most people got interested in the study of apoptosis. Uh, historically, there was one lineage of studies which stemmed from developmental biology, which focused on the phenomenology or describing the phenomena which could come under the umbrella of programmed cell death or cell suicide or apoptosis. And I'm not going to get too hung up with the fine distinctions of cell death which may come under this umbrella, but very roughly speaking, it can be described as the phenomenon in which a cell kills itself or is made to kill itself by delivery of agents which make it kill itself. And in many ways, this is a conundrum. But from the developmental viewpoint, it was seen as a mechanistic process in, say, sculpting the fingers of a tetrapod hand, since ultimately the tetrapods have emerged from marine ancestors, which had fins of flippers, if you may want to call them that, like the lungfishes and the coelacanths, which are the sister groups of the tetrapods. And they are bona fide aquatic animals which swim around with their fins. And from that fin state to the state with fingers meant that the connective tissue or the tissue in between the bones supporting the ancestral fin, which was to now give rise to the hand of the tetrapod, had to go away. And developmentally, that process happens through cell death. And you can observe this very clearly through a very common technique which we used to use in college. That's the study of the chick development, the chick embryo development, where we used to make these slides to observe cell death in the interdigital regions. So for the developmental biologist, it was an essential process of animal development. For the plant pathologists, which is the background you originated from,
Right.
it was again a very commonplace phenomenon, a response to viruses, hypersensitivity response, as it may be termed, viruses and bacterial pathogens, where cells kill themselves to limit infection. And eventually, a similar kind of phenomenon came to be recognized in vertebrate immune systems, where you have a programmed cell death of this kind. But so these were the broad lines of research encompassing organismal biology, developmental biology, and cell biology, which made the phenomenon interesting. But from my own perspective, that was not how I got interested in it. It was from the evolutionary side. It goes back to a question which fascinated Darwin himself at the foundation of what we could term modern biology. We can sort of place a boundary in the history of biology that since Darwin and Wallace's work presenting the framework of evolution or the evolutionary theory, we have had real biology or biology with a theoretical foundation. And one of the issues which came up right in that theoretical foundation is why do organisms cooperate? More generally, the question of altruism. So you see altruism all over nature. Humans, for example, exhibit altruistic tendencies, and Darwin was very aware of that, and it puzzled him because from the more essential version of the evolutionary theory, an individual is trying to maximize its fitness. So if an individual sacrifices its fitness for some other individual, it makes no sense as to why this trait would persist, for natural selection would eliminate it by definition, since the fittest survive. And if an organism is nullifying its fitness, the whole question arises as to how a trait which causes fitness to be nullified in order to enhance the fitness of some other organism in the population gets selected for in the first place. And this became even more apparent as people studied social organisms, eusocial organisms to be more precise, like hymenopterans, it's the insects or termites, which are related to the cockroaches. And you have within that isoteran, dictyoteran clade of insects, cockroaches, termites, praying mantids. Praying mantids and most cockroaches are not social. Each one is maximizing its own fitness as an individual for most part. And then you suddenly get these termites, where you have a very well-developed sociality which closely mirrors human society, with a king, a queen, and various castes which are specialized in terms of their labor, close parallels to human society. And the queen is the reproductive female there, the king, the reproductive male. And the other individuals have forsaken their fitness for this, for these two apex members of the society, so to say. So that is a big puzzle in itself from the older, the more essential form of the evolutionary theory, which Darwin and Wallace presented. And there has been a reflection of this amid social thinkers over the ages, like in our own tradition, where there's been a great emphasis on social cohesion through division of labor and interaction, a synergistic interaction between groups specializing in some form of labor. So why should this kind of unit exist? There has been some thought about it. So it is a long-standing question in biology, going even back to the times before the Darwinian framework for biology came into being. And perhaps the first person to address this with a very pithy solution was the great Jabius Haldane, who said, who you would recall his famous statement. He said something like he was willing to die for a couple of brothers or for 8 first cousins.
Right.
So, yeah, so essentially that gives you the answer. You have you share 50% for a diploid sexually reproducing organism who with full siblings, that is descending, the siblings descending from the same pair of parents, one would share 50% of the genetic material with them. So if one were to die in order to foster the fitness of 2 such siblings, then in a sense your fitness is not going in vain, even though it by itself would be 0. You're getting a gain via those siblings of yours. And likewise with your cousins, in which case there's greater dilution, but through 8 of them, you could make up for your own loss of fitness, as long as that, especially if that goes into contributing to their survival. There's a situation where if by dying yourself, you're going to let 8 of your cousins or two of your siblings further their genes,
right.
then it's a net gain. There is no loss in your death at all. And that was the thing which Haldane so pithily expressed. The interesting thing is people ignored this thing of Haldane. One would have thought that it would have sparked a whole revolution in the understanding of or the very foundations of the evolutionary theory, since it was answering an important question which had puzzled Darwin. But it, like many discoveries, including some of our own, they are quite forgotten when they are made, and they pick up steam many years later when someone else rediscovers it.
Right.
And in this case, it was William Hamilton, who overall was quite a genius. And in many ways, like Haldane, he was a pretty remarkable thinker in terms of evolutionary theory, and also an engaging writer. Both of them were engaging writers. We have read the essays of Haldane and found much of interest in them. He was
Right.
always an acute observer. And that applies to the fertile imagination of Hamilton too. And Hamilton formalized these ideas, bringing forth the concepts like kin selection and inclusive fitness. So, just to expand on these terms a bit, inclusive fitness uh, is the idea that it's not the propagation of the genes which you carry alone which contributes to your fitness, but you can gain additional fitness through your siblings, through genetically related individuals. And the coefficient of relationship, which is a measure of how much of your genetic material you share with those relatives, would essentially give you that the multiplicand, or it would give you that number which you can multiply to uh, measure how much fitness you gain via them. So if it's 0.5 with full siblings, with two siblings, you can make up one of yourself. And kin selection is a related or a consequential term. It's a term which stems from this, that it's not, selection is not merely acting on the individual, but also on account of this inclusive fitness on the kin. And if there are behaviors which are going to help the kin prosper, it means that prosper in a very strictly evolutionary sense. That is, it's going to help them increase their fitness at the cost of your own fitness. You can set up a very simple cost benefit inequality, wherein the cost to your own fitness uh, subtracted by the gain via your kin, multiplied by the number of kin you're helping to propagate in terms of their genes. That inequality will give you a measure of how much fitness you're going to accrue via your kin. So it's a simple cost benefit inequality, which would allow you to estimate where selection would tilt. So if a behavior is going to cause self-sacrifice and it is going to cause the survival of the kin, then if it's going to be of the type that on your own, say you're going to make only one copy, but through your self-sacrifice via your kin, you're going to make many more copies, like say 4 copies, and those kin are fully related to you. That is, they are genetically identical to you, which would be the case with unicellular organisms like bacteria, which are clonal. Then what you would see is a selection for that self-sacrificing behavior due to the inclusive fitness gained via the kin. And this was a very profound idea when, even though it's simple but profound when Hamilton popularized it. With Hamilton, it received much greater attention than when Haldane first put it forth. And in part, that was due to the hymenoptera, which we spoke about.
Right, the haplodiploidy.
Right. So the peculiar haplodiploid system, which meant certain kinds of relationships between siblings and parents and siblings. And that was taken up by E.O. Wilson, um, the famous hymenopterologist, who presented evidence that indeed kin selection was likely operative and probably responsible for the emergence of eusociality in organisms like ants and bees. So it was, in a sense, the test of the hypothesis of kin selection, and the hypothesis seemed to survive the test. Though I must remark that in recent years, Wilson has backpedaled, uh, and he seems to favor this broader idea of selection rather than kin of group selection.
Mhm.
And that is a somewhat vaguer concept, but we can't rule it out entirely, that perhaps these self-sacrificing behaviors might emerge even within groups, especially if groups are in competition with each other, or whole lineages of organisms are in competition with other lineages. Uh, so group selectionist or behaviors, self-sacrifice behaviors favoring the entire group might come into play. And it's possible that such behaviors have played a role in human society, in the emergence of human societies where there were whole bands of, say, hunter-gatherers or farmers competing against each other, uh, where perhaps there was some room for a group selection model to emerge. And in the case of Wilson, he argues that that may be true even for hymenopterans and the isopterans, that's the termites, uh, wherein, at least to initiate uh, the process of sociality, there may have been a role, and eventually he posits a role even in its maintenance, but I guess we don't get into that. That's just the background. For our purposes, I think kin selection uh, is more than sufficient. So, anyhow, to cut a long story short, it was these reflections on kin selection in the early 90s which made me realize that there's likely to be some very deep connection between three things. One is the emergence of multicellularity. The second is infection by pathogenic entities or parasitic entities. And the self-sacrifice of cells, which essentially is what we term apoptosis.
Right.
So that is approximately how, the course of how I reached this point, or I reached the point of being interested enough in this phenomenon that it was going to offer us a window into understanding these very profound questions, such as skin selection at the molecular level.
Right. I mean, the interesting thing is that whereas at the molecular level, and the consequences of which we shall see in the following sections, it's a principle operating with proteins functioning in a certain way. But in general, as such, say, when you look at kin selection, say in a hymenopteran context, in a hymenopteran society, it's the principle that operates, and it may not have anything to do with the proteins involved or similarities in that. There, it could be a whole other range of components that are causing that.
Yes.
Behavior, right? So it's something that emerges under the consequence of a large number or a fair number of closely related or identical cells or organisms or units working together.
Right, right. So essentially we can see this as a derivation from first principles of biology, which is the evolutionary theory. So as long as we have the very natural extension of the Darwinian theory to include kin, the concept of inclusive fitness and kin selection, then a prediction made by that in microbial or at the cellular level aggregates of cells would be the existence of apoptosis. And it parallels what you see at higher levels or larger units of biological organization, like a eusocial society. And that ties in with the idea of the superorganism, which has often been used by biologists to describe eusocial societies. So the whole conglomerate or the colony of ants or the hive of bees, they are seen as one organism or the superorganism. So if you reduce it down, you can see the lower level or at the cellular level, the same principle in the form of multicellularity and apoptosis having an interplay with each other. So you can, in a sense, there is a scaling of this principle from cells and aggregates of cells, eventually leading to one organism, and whole organisms cooperating into a superorganismal assembly. So, right. Yeah. So, and perhaps it goes even further down at the level of the genome, because at the end of the day, each gene is, in a sense, in cooperation with other genes in the genome. So, a gene has two options. It can break out on its own and further it copies of itself, or it can cooperate with other genes and help the furtherance of the genome as a whole. And there is a certain balance. There's again a cost-benefit analysis. For most genes, breaking off on their own is not an option. But some genes have specialized in that breaking off of their own, most trivially in the form of transposable elements. Yeah. So if you have an endonuclease or a polymerase, which can somehow catalyze making more copies, either directly or by using the cellular replication apparatus, then you have the option of breaking out on your own. But there is also the issue of freeloaders. So if you freeload too much on the system, you could cause the whole system to collapse if you're not cooperating. So there is a certain balance which is reached between breaking off of your own on your own as a transposon and contributing something to the organism. And thus we have a whole bunch of elements, which was one line of study which has dominated the kinds of things which we have done so far, are those which are on the twilight. Transcription factors, for example, they are domestications of transposons. And the whole collection of these immune systems at the molecular level, the cellular immune systems, the restriction modification systems, the CRISPR-Cas systems, and more recently, these nucleotide-dependent effector systems, they can all be seen as elements which are at the twilight of selfishness and contributing something to the cell, and they reach a certain balance of cooperation. In terms of sacrificing their own fitness of making many copies to a degree, and helping the conglomerate of genes termed the genome to survive. And there are one group of elements, the toxin antitoxin systems, which are even more ambivalent in this regard. And that we can talk a little more of that going down the line, because it does relate to the story of apoptosis, of trying to understand apoptosis.
Right, right. I mean, and I think that is the general purpose of this conversations, or these set of conversations that we're going to have, which is to sort of stitch the whole thing together, to give a more universal perspective that we hold of
Right.
these processes. And
Yes.
in this regard, I think you bring up a point that we've often talked about, over all these years. That is, the general laws of biology. I know it's one of your pet topics, and I think, in a sense, you have described this aspect of kin selection as a general thing that emerges in biological systems at different scales of events.
Yes. Yes. Yeah, I think one other point which we should, so there are two principles, two very uh, extensive principles when we talk of laws, to the extent we can talk of laws in biology. And there's a very deep philosophical point here, which I could just put it out there, because I don't know for certain the answer for it. The philosophical point is whether these so-called laws or principles, they are completely emergent, in the sense we can predict them. If we know all the physics, then we can predict all the chemistry. If we know all the chemistry, it would encompass all the biochemistry, and hence these laws just automatically fall out of biochemistry. Or are there some more general principles coming outside of physics and biochemistry which impinge on these? My own thinking is that indeed such principles exist, and that's where these so-called emergent laws of biology are. So are they emergent purely at a higher level from the physics and chemistry, or are there some pure constraints which lie in some Platonic realm, just like geometry? So geometry is one of them. As we have often discussed, Euclidean geometry is a very profound foundational principle from which one could derive a whole set of higher order principles which are foundational to biology. For example, the very reason that you have evolution through natural selection perhaps boils down to principles rooted in Euclidean geometry. We'll not go there for now, but I'll just put it out there. And then there is a more general version of that class of Platonic principles, which may be termed linguistic. And as we proceed into the molecular accounts, we may encounter some of these, like when we describe architectures of proteins in terms of their domains and the like. But more generally, I would like to point out to two principles, which may be termed as some kind of duality. To use a very rough analogy, which should not be taken as anything more than some kind of imagery. It's not an analogy of purpose or intent. It's like in physics, we have the wave-particle duality, where you may describe a very small physical entity as both a wave and a particle. It's both things at the same time, and depending on the experiment, you may see something looking like a particle or looking like a wave. So likewise, there is a duality with respect to this whole concept of multicellularity and unicellularity. We may say that ultimately there is neither pure multicellularity nor pure unicellularity. Every organism is, in a sense, a superposition of both these states. So what I mean by that, a microbiologist would easily understand this. If you put a cell, an E. coli or Bacillus subtilis on your Petri plate, uh, it grows to give you a very visible colony. So so does that mean that E. coli is a unicellular organism or a multicellular organism? That looks very much like a multicellular assemblage. So the very fact that you have a boundary condition in the form of a cell surface which separates two cells means that upon cell division, you're going to have two kin cells
Right, right.
adjacent to each other. So by definition, you're going to have an aggregation of kin by the very definition of life itself, and hence the process of cell division. And in many cases, that is a state of existence, like biofilms, where it could be a mixed biofilm, like on the tooth of an organism with a very wide diversity of bacteria on it. But but still, locally, it will be a collection of kin who have come from the cell division, repeated cell division of an ancestral cell. So multicellularity and unicellularity, they are not hard and fast states, and you can alternate, or alternate may not be the right term. More precisely, you can say you can have episodes of multicellular and unicellular existence. And sometimes this may be formalized at some level, like say a bacterium like Bacillus subtilis, it may have a substratum, a substratal growth, and those which are more to the top of the colony, they form endospores, which are then dispersed by phenomena like water and wind and so on. And they found new colonies which propagate this system. So at some level, Bacillus subtilis exists as both a multicellular and a unicellular organism. And we could say that even of things like plants and animals, which we normally see as multicellular, many of them have gametic states which are purely unicellular. Right. So this is one duality. And an important corollary to this duality is that there is a very good chance you find yourself in the neighborhood of your kin. And finding yourself in the neighborhood of your kin implies that forces such as kin selection are going to be very important in the evolution of any organism. And another such basic principle to keep in mind, and this goes more into the molecular level, is the idea of the emergence of dual functions or antagonistic functions. So this needs a little more consideration, or it has to be explained in some more detail. Now imagine you have an enzymatic activity which has emerged in a protein. And that enzymatic activity has some kind of a deleterious role on the cell. So how would you imagine that being fixed in the first place? Because it is going to there's going to be a negative consequence for that. Now, if a mutant version of that were to emerge, which were to not have the deleterious activity, perhaps be inactive entirely, and there's a tendency for proteins to dimerize. It's a very widespread tendency. Multimerize, dimerize at the most basic level. So what could happen is that deleterious activity gets masked by this inactive paralog emerging, because there is some kind of a interaction, the basic dimeric interaction is preserved, and there is a kind of a dominant negative effect, wherein one suppresses the other. So you can imagine the emergence of what can be termed suppressor mutants for a deleterious gene. So whose products are, so you have two paralogous versions, one with an active deleterious role and the other which is essentially inactive but suppressing the deleterious role. And in genetics, we always use these tricks to get suppressor mutants to understand function. And since both of us are well familiar with doing some genetics with our own hands, we are perhaps that last generation of computational biologists. I don't know, maybe the newer generation is more into doing these old style experiments. But suppressor mutants readily can be obtained.
Right.
So the principle of these suppressor mutations and the existence of suppressor of a deleterious gene and its suppressor implies a certain masking of the deleterious function. So this is of great importance when we understand, when we try to apprehend something like the origin of apoptosis. That the emergence of suppressor mutants or suppressor mutations, especially in the form of paralogs, a deleterious paralog and a suppressor paralog. But it need not be so. It can be in the form of, say, an enzyme which has a deleterious activity, and it gets suppressed by a transcription factor, which is the emergent suppressor mutant, which is now able to bind its promoter region and block its expression. Right. Or a protein which, the suppressor mutant, which emerges, which physically interacts with the deleterious protein and thus suppresses its deleterious effect. So this principle of the emergence of suppressor mutations is central to the emergence of this process of apoptosis, because we need effectors which will kill the cell. And one of the big evolutionary conundrums is how do these deleterious effectors which are going to kill a cell emerge? So to summarize, it's a collection of these thoughts which led me towards apoptosis. First was thinking about the basic puzzle which Darwin faced, emergence of altruism, and extending that from the level where it was normally studied, like hymenopteran colonies or termite colonies, down to the level of cells. So in the early 90s, that was one thing I was thinking about. What was the consequence of this when we extend it to cells? The second was this idea of the duality, that there is no truly unicellular or purely multicellular organism. It's a state of duality, of having both multicellular and unicellular states. And a corollary to that is that kin selection is likely to be important at some point in the life of an organism, because by the very act of cell division, which is the definition of life, you will be surrounded, or you would be in the company of kin. And the third was this was the general thinking which I was doing about suppressor mutants. So from the first time I studied genetics and learned of the existence of suppressor mutations, it led me to think about its consequences. And one thing which became immediately apparent was that it could allow the emergence of balanced deleterious activities within the cell. So essentially, you would have a pair of proteins or a pair uh, of biopolymers, typically two proteins, but it could be a protein and RNA too.
Mm-hmm.
Uh, or it could be some genetic element even at the DNA level and the protein, which one of which is deleterious to the cell and the other is suppressing this its activity. And then you have stabilization. So you have both of them existing. It's a cancellation. Uh, Thinking more about this third thing led me to realize that you could thus have these potential complexes. And here I'm using complex in a functional sense, uh, where you have two interacting entities, biomolecules, where one can harm the cell and the other can suppress that harm. And if there is some kind of imbalance which propagates through the system, which causes that suppressor entity to dissolve away, then it can unleash this deleterious activity which can kill the cell. And there may be special circumstances where this kind of behavior can be selected for, especially when you place it in the context of kin selection, where the deleterious activity is unleashed in a cell and the cell dies, but that death proves to be advantageous to the kin who are naturally surrounding it as a consequence of the second principle which we just spoke about, which is the you tend to exist with your kin, you're coexisting with your kin. So thus you can imagine a molecular pathway or the molecular mechanism for the emergence of altruistic behavior. So this was sort of, I thought of all this at a theoretical level, maybe by around 1991, 92. And that's what formed the platform, or it was the foundation when, now having thought of all this, I decided to go out into sequence space and look for them.
Right. So I mean, there were two great things that happened in the sense that the emergence of being able to actually find out the protein components of these processes. By the 90s, we were able to get a protein sequence, proteins that were involved in this process, and across different forms of life, and also the emergence of databases of protein sequences.
Right.
We could sort of now do a comparison to see what is conserved across life or across whatever is there. So I think that will be the basis for our next section. I think we can stop here because this is a good point where we've covered the basic theoretical background for why study apoptosis, or what were the interesting aspects that led you to even sort of theorize about apoptosis or kin selection in general.
Yes, I think it's a very natural progression because, as I said, in a sense, that's how it worked out for me. I was thinking a lot about these theoretical issues, and I thought, okay, now the theoretical issues lead to some predictions, and we should be able to test those by going out into sequence space. And the early 90s were a good time when sequence space was expanding, and we could actually do that as long as you knew what you were looking for. And that story took off in three directions, which have actually been very big directions for my research ever since. As you can imagine, one of those places, I realized that it was already there, in the form of restriction modification systems.
Right.
Because here you have two kinds of activities which are opposing each other. And these have been known for a very long time, going back to the pioneering work of Werner Auber and then Hamilton Smith and Meselson, some of the founders of modern molecular biology. Um, Rich Roberts, subsequently his extensive work on these systems. And it was reading, it was one of the reviews which Rich Roberts had written, which sort of made me realize that, hey, we are sitting on this kind of the pair, the deleterious enzyme and its suppressor enzyme right here. And I think it was Kobayashi's work which showed that once you lost the methylase, you could have death of the cell due to the restriction enzyme. So right there, we had evidence that such things existed, and that provided a template for what we may look for in terms of the emergence of such systems. And that was also the period when eukaryotic studies on apoptosis were entering the molecular era. So one could tie in the observations coming from there with these observations on the restriction modification systems. And the third wing which came in at that point were the toxin antitoxin systems, which we can talk about a bit in the in the coming discussion.
Right. Yeah, but just as a aside, I don't know if you wish to talk about it, but I have seen your extensive notes, and I remember your earlier days were filled with periods where you would physically copy down the sequences and then enter it into a database of protein sequences that you had to do your comparisons. I think we've come a long way from there. Unfortunately, we don't have to do such anymore. But I do think that
Yeah, those were sort of the testing, the days of the testing ground. It was GenBank would always arrive sort of like 6 months late in India in the form of CDs. At least the place where I was.
I remember that.
So we would have a lag of sequences by 6 months. And so the only way was to physically copy those sequences in the library, since there were no Xerox facilities in several of those libraries. So you had to actually write down the sequence and then key that into the local database which you maintained. That is the self-updated GenBank, so to say, or the self-updated NR to be more precise.
Right. Oh, well, I mean, that's a little bit of personal history.
Yeah.
But okay, so let's stop here for now, and let's in the next section we'll discuss more on the molecular aspects of this and the early history of the discoveries of the proteins involved in this.
Yeah.
Okay.